Hello, my name is Sarai, and welcome to my presentation, A Characterization of the Microbiome of Schmidtea mediterranea. So Schmidtea mediterranea is a species of planarians, which are flatworms naturally found in freshwater aquatic environments. They're most known for their ability to regenerate from body fragments to full adult animals. This makes them perfect model organisms for stem cell research. Currently, I am an undergraduate research student in the Pelletieri Lab of Keene State College. So this is where I got the idea to use the planarians as a source of sample. So not much is known about the microbiome of planarians, which I found very interesting and drove my curiosity. So the purpose of my project was to culture the readily available microbial life associated with the planarians. Current research for the microbiome of planarians is very low that I was able to find. One study used some 2-week starved planarians housed in laboratory conditions and were able to grow microbial life off of those worms. It found that most of the phyla present was the proteobacteria, as well as had some firmicutes. Most were gram-negative, which makes sense if most are the proteobacteria. And the largest genus found were the Pseudomonas. They also studied bacteria that were occupying different areas of the worm. For instance, bacteria found in the gut versus the mucus and both. So to start off my project, I did some preliminary sampling and cultures. First, I started with 3 tubes of tryptic soy broth and placed 5 live worms in each tube and left them overnight to shake at 150 RPM at room temp. After 24 hours, I did not see any growth, so I removed the now dead animals and placed the tubes back on the shaker. After about 72 hours, I was able to get some growth. So for each of the 3 tubes, I aliquoted out 100 microliter samples and placed those into 2.5 mils of TSB and placed them in the shaker overnight, this time at 200 RPM at 37 degrees C. Finally, after 24 hours, I plated the liquid cultures using just a sterile cotton swab and streaking it across a tryptic soy agar plate and left them in the incubator overnight at 37 degrees C for 24 hours. This next slide depicts what I just explained in the previous. So figure 1 shows the live animals in the broth. Figure 2 is the 6 aliquoted samples I was able to grow overnight. And then from each tube, I was able to streak them across some TSA, grow them, and then this is what I use to collect biomass for DNA sequencing. So for my data, I have 2 charts here. Figure 4A shows the overall community DNA sequence data. As I expected, most was bacterial life, adding in some archaea, eukaryota, viral. Of course, there's a portion with no hits, and then a large of the other, which is a part of the root. 4B depicts the top phyla present in my sample, which compared to the current research, a large portion of mine was also protobacteria, followed by the firmicutes, and then the actinobacteria, and then all other phyla. This table depicts the top hits that I had for my sequencing data. The first column represents the percent coverage, so how many hits. The second is the rank. So for example, bacteria will give you a D for domain, so on and so forth, all the way down to an S for species. The top species that I had hits for were the Stenotrophomonas montifilia and the Staphylococcus conat. So starting with a little information about my microbe, the Stenotrophomonas multifilia is a gram-negative bacillus, closely related to the Pseudomonas. S. multifida is an environmental pathogen and can be found just about in any environment, including animals, plants, soils, and clinical settings. This is a human pathogen that's an MDRO, or a multi-drug resistant organism. So it is very hard to treat. It can cause severe infections in immunocompromised patients, such as HIV patients, post-op patients, even long-term care patients. Some of the associated infections include UTIs, which you see a lot in long-term care patients. You also have endocarditis, meningitis, and pneumonia. One fun fact that I also found was that S. multifida has been shown to adhere to plastics and biofilms, which I found interesting in a clinical setting aspect, because that's something that we don't want, especially, tubings or instruments that we're using, say for cleaning up wounds, or, for instance, a UTI that could be caused by catheter insertion, things like that. So the second part of my project was to get down to as pure of a culture as I can. So this is going to be the second half of the presentation. So here I have one of these street plates that I was able to grow some bacteria on. I was able to get down to some individual colonies, and here I just wanted to go over some of the colony morphology. So they were white, opaque, circular, with very smooth edges all around. I also did some gram staining of the culture. And as you can see here, we have a nice purple color, which indicates that they are gram positive. They also are very circular around, clustered together, giving them a nice staphylococci arrangement. So my candidate for the pure culture was the Staphylococcus coni, which is a gram-positive, non-aureus staphylococcus. It's commonly found on human skin and on dairy farms, which we actually feed our planarians in the lab calves liver. So I found this very interesting. A recent study shows that they may act as a reservoir for antimicrobial resistant genes, or AMR genes. These genes are coding different types of proteins that will combat against antibiotics. For example, antibiotics such as beta-lactams, so your penicillins, amoxicillins, AMR genes can code for beta-lactamases that will actually degrade these antibiotics before they have any sort of harmful effect on the bacteria. So in this paper, they did find a number of isolates that did house some of these AMR genes. So along with the beta-lactams, you also have things like the aminoglycosides, which gives you your streptomycin. So due to that study that I found, I wanted to test antibiotic resistance on my microbes. So I performed a Kirby-Bauer test. As you can see, my microbes are susceptible to all of the antibiotics used when compared to the control. So to wrap up my experiment, I wanted to think overall what I would do different next time if I did repeat this. One thing is to use multi-week starved animals. I'd like to make sure that the microbial life that I grew from the worms is coming from the worm itself and is not so much influenced by what we feed them, which is the calf's liver. I'd also like to look at where the microbial life is occupying the worm, so the gut versus the mucus versus both, as well as how different housing conditions can affect the microbiome. So for instance, leaving them in static water conditions versus, continuous filtration. I believe that more research could be done on S. coni to further research into AMR genes. Antibiotic resistance is plaguing us, as you know, and after our current situation with the pandemic, I believe this is such an important field and is something I would like to go into myself, as well as bring it all back to the worms themselves. I would love to see in the future planarians being used for studying immune responses to MDROs. I'd like to give a thank you to the Hubbard Genome Center of the University of New Hampshire, which is funded by the New Hampshire Embry program, for doing the sequencing data for my project, as well as Keene State College. I would like to thank Dr. Lauren Lonan for guiding me throughout the semester with this project and kind of helping me bring everything together into a really solid conclusion. Here are my references for my presentation, and I hope you enjoyed it.